last talk of the day, even though I am standing here. And Clara, who's on screen, knows that I absolutely detest Zoom and under no circumstances allow anything to happen by Zoom. But Clara gave me permission to say, in order to defend myself from anybody ever asking again to go on Zoom, that she's had a recent medical issue and she would have loved to be here, but she can't. And we also offered for her to just cancel, but she wanted the distraction. And Clara is a fantastic speaker, doing fantastic work. She used to be here at UC Berkeley with us, and she's now been, I don't know for how long, I think you can correct me if I'm wrong, I think you're in the frontier subgroup of prescient design within Genentech, like some Russian dolls, but I'm going to hand it over to you in a second. But these talks have been very interactive, and I suspect, Clara, you would enjoy interactivity. So we'll have to kind of figure out how to do that. If you want to periodically pause, we can, as long as we hand the mic around, then you'll be able to hear us. So feel free to kind of do what you want, and we'll move the mic around. So yeah, sound good?
Yes, that sounds great. Awesome. Okay, yeah. Hi, my name's Clara. As Jen said, I am a research scientist at the Russian doll of frontiers within prescient within Genentech. And the talk I want to share with you guys today is called Error Embraced, Making Trustworthy Scientific Decisions with Imperfect Predictions. And really want to give a huge thanks to the organizers, Jen and Aditi, for inviting me to do this, and also the staff at Simons. I'm really, really jealous of all of you guys in the audience who've been able to be there the whole week. But like Jen said, I had a series of medical emergencies that kind of made different plans for me. But I'm happy to be okay and still able to do this over Zoom. And like Jen said, please interrupt at any time with any questions. I think I have a decent view of the audience, so if you just like do something dramatic, I can probably see and stop. But if not, get Jen's attention or something. I would love to answer any and all questions. I don't have a packed agenda for this talk, so please feel free to interrupt at any point. Okay. So I'm actually going to start this with a little game that I think this audience might be very adept at. So here are two protein structures. One of them is an experimental protein structure, and the other one is the AlphaFold 2 predicted structure. Does anyone have a guess for which is which? It can be a not informed guess or informed.
One says the right one. John Ingraham in particular says that the right one is AlphaFold. Which version, John? Yeah, okay.
All right, so we have a vote for the right one being AlphaFold. Any other guesses?
A second.
A second confirmation for that. Okay.
John is swaying the crowd.
All right. Okay, well, so it turns out that actually the one on the left is the AlphaFold prediction, and the one on the right is an experimental NMR structure. I mean, it's kind of people kind of assume I'm saying crystal when I say experimental, but yeah, I guess like the point is really just that it's not always straightforward to tell, even though the AlphaFold prediction does get some very important parts wrong. And so the high level motivation of my work and the method I'm going to be talking about in particular is that as a scientific community, we're increasingly using machine learning methods and the predictions they provide as basically inexpensive, high throughput proxies for scientific experiments. So that includes, of course, AlphaFold. That includes models that predict gene expression or variant pathogenicity, models that predict diagnoses and pathologies from medical images. And we're all very excited about using these predictions to basically accelerate the process of science. But like, as we all know, predictions contain errors. And so when I draw a conclusion using predictions, how do I know I can trust that conclusion that I draw? So if I draw some conclusion based on an AlphaFold predicted structure, how do I know that that conclusion wouldn't have been different if I'd basically been looking at the kind of analogous high quality experimental structures instead? So that's essentially the motivating question of this talk. How can we use machine learning predictions to make trustworthy scientific conclusions? And stylistically, this is not going to be about ways to train particular models or very specific architectures or whatnot, but rather, given that you have a promising predictive model for your domain, no matter what that model looks like or how it was trained, how can you then use its predictions in a kind of provably reliable way? So this is actually work that's titled Prediction-Powered Inference that I did with these incredible colleagues here, Anastasios Angelopoulos, Stephen Bates, Mike Jordan, and Tiana Zernich. And this was actually work done when we were all at Berkeley about a year ago. So I'm going to start again with a motivating story about proteins, because it really did kind of kickstart our adventure here. As we know, a protein is a sequence of amino acids that folds, or at least most of them fold into some sort of 3D structure. And studying features of either the sequence or features of the structure can give us lots of insights into protein function or disease mechanisms or even medicine. So as an example, in 2004, biologists made this really important observation that post-translational modifications, or PTMs, and these are chemical modifications to the sequence that regulate the protein's function. It seemed that these occurred quite frequently with intrinsically disordered regions. And that's just a region of the sequence that doesn't really fold into a fixed 3D structure. So this was a very insightful observation about 20 years ago, and it went on to motivate lots of lines of work in biology. But back then, back in 2004, they couldn't actually reliably quantify and put a number on this association between these sequence and structural features, the PTMs and IDRs. And that's just because there weren't enough high quality experimental structures for them to kind of compute any sort of value. Now, fast forward 20 years, almost 20 years, by 2022, we have AlphaFold predicted structures for hundreds of millions of protein sequences. And then that same year, a study came out that used those abundant predictions to kind of finally quantify this association. And they did that by computing an odds ratio between the presence of these two features. So an odds ratio between the presence of a PTM, post-translational modification, and the presence of intrinsically disordered regions. Okay, and the twist here, though, is that the study wasn't actually looking at intrinsically disordered regions. They were looking at predicted IDRs based on the AlphaFold predicted structures. And this, the entire work was kind of built around just using these AlphaFold predictions to compute these associations and make conclusions about these kinds of features. So this points to a bigger story happening all over science, which is that predictions are actually being used to ask and answer scientific questions, draw conclusions about the nature of reality. And this is happening like not just, in the protein sciences, but there are studies now using this to study disease-causing variants, regulatory DNA, the demographics of patient phenotypes, and even beyond biology to studying human welfare more generally, using data that's like not really data as we would typically define it, but using predicted quantities. And in many ways, it makes sense to be doing this. We all know gold standard experiments are really expensive. They're really time consuming. They require highly trained postdocs, lots of grad students, whereas predictions are very cheap and instantaneous, and they're really easy to get. Despite that, it should make all of us probably a little bit uncomfortable if we're going to be drawing conclusions based on predictions, because of course they're just not the same thing as experimental, kind of gold standard experimental results. So for example, in the paper that we just looked at that computed an odds ratio, if it gives me an odds ratio of like 1.5, how do I know that 1.5 is real? How do I know that it wouldn't have been 0.5 or some other completely different value if I'd been looking at the experimental structures instead? So prediction-powered inference is a framework for doing provably valid statistical inference when you're using predictions as if it were data. So on this page, or on this slide, I'm just going to lay out the general problem setting that we're trying to address. So the general problem setting here is that we have some labeled data consisting of features X and the label Y. And Y is anything you would typically want to measure with a gold standard experiment. So for example, like X could be a protein sequence, Y could be a structure or a feature of the structure like we just looked at. X could be any other kind of biological sequence, Y could be a phenotype, X could be an image, Y could be some label of that image, lots of other examples. And in addition to this labeled data, which I'm also going to sometimes call gold standard data interchangeably, we also have a much larger set of unlabeled data. And that's kind of indicated by the length of those bars. And that unlabeled data also has features X prime, but here the labels Y prime we don't have access to. We never ran those experiments, or those labels are inaccessible. So for example, this unlabeled data could be all protein sequences that I might be interested in for a certain scientific study that I wish I had structures for, but I just don't. It could be a huge database of medical images that I wish I had kind of gold standard pathology labels for, but I don't. Or there's a number of examples that we can kind of think of that fit this setting. And importantly, for both the labeled data and the unlabeled data, I have predictions that I'm going to call F and F prime made by a machine learning model. And throughout, I'm just going to assume the model is already trained on separate data. So the model that provides F and F prime is trained on some data that has no overlap with the labeled data, no overlap with the unlabeled data. So it's just some like separately trained fixed prediction system that I'm just going to use to make predictions for both the labeled and unlabeled data that I have here.
There's a request for a question.
Yes.
How important is it that F is trained not on this particular labeled data?
Yeah, that's a great question. So the labeled data we're effectively going to use it to quantify the prediction error and how it will affect the quantity we're going to estimate. So if you did have overlap, it kind of be like double dipping, essentially, like you wouldn't get an actually correct evaluation of the prediction error, and you wouldn't kind of get provably valid inference. Does that answer the question?
We got a thumbs up.
Okay. Thanks. Yeah, please feel free to interrupt if there's anything that's not clear here. Okay, so we're going to focus on a common setup in basic science, which is just that there's some estimand of interest that we want to know the value of. And this estimand is going to be some property of the underlying data distribution. So for example, it could be the mean value of the label Y, some quantile of Y, and it could be regression coefficients between Y and the features X. So just to instantiate that a little bit more, the mean value of Y can very often be used to capture like the frequency of a certain event or a certain phenomenon in a population, in a distribution. Regression coefficients, of course, can allow you to study associations between different labels and different features like disease and demographics, species and temperature. So these are just some examples. So the goal of our method is going to be to construct a confidence set, CPP, or prediction powered confidence set, that is valid. And by valid, we just mean that the estimand, which I forgot to mention, we're going to call theta star, the estimand is going to fall into that confidence set with guaranteed high probability. So it'll fall in that confidence set with probability at least 1 minus alpha, where the user can specify whatever alpha between 0 and 1 that they would like. Okay, so how might you achieve this goal right now with existing tools? On on one end of the spectrum, in what we call the classical approach, you could just use the labeled data and totally ignore the predictions. And this is basically how science has been conducted since the birth of modern statistics. You just have some gold standard labeled data, you work with that data, and you make some conclusions using kind of existing statistical tools. And there's like a whole wealth of existing statistical tools people use to construct confidence sets that are valid or that have kind of similar guarantees. And so this is great, you get valid results, but you also totally lose out on the information that is contained in the predictions. So, like the reason why we're all in this room or virtually in this room is that predictions are pretty good. And even when they're imperfect, there's often such an overwhelming amount of unlabeled data, overwhelming amount of predictions we can get that if you just totally throw that out the window, you're missing out on some kind of evidence you could be using toward estimation. So that's like the downside of the classical approach. You're throwing out information. On the other extreme, in what we'll call the imputation approach, you could just imagine treating the predictions as if they were data, as if they were like gold standard labels, and being blissfully merry and carrying on with your day with existing statistical tools. And if you do that, you will get confidence sets that are invalid because the predictions contain errors, as we know. And so prediction-powered inference is just a framework for using both the unlabeled data and the labeled data to get you the best of both worlds. We're going to have confidence sets that are provably valid while still squeezing information from the predictions to effectively increase your sample size. So practically speaking, what that means is you're going to get smaller confidence sets than you would have if you had just completely ignored the predictions. Okay, so this is like the general setup of the problem and what our goal is. I'll stop here in case there are any questions about just the setup for what we're doing.
Perfect understanding in the room.
Okay. All right, great. So before I go into how this actually works, I'm going to show a quick demo from that same paper that used the AlphaFold predictions to compute an odds ratio between PTMs and IDRs. And just briefly, as a refresher, an odds ratio is just a way to quantify association between two binary events. So here, the binary events are the presence or absence of post-translational modification at a sequence site and the presence or absence of intrinsically disordered region at that same site. So if the odds ratio is greater than 1, that means that those two binary events are basically correlated. And if it's less than 1, then they're like effectively anti-correlated. So that's just for some context as to what this x-axis is. So for this demo, we looked at the portion of their data that did have gold standard labels of intrinsically disordered regions. And on that data, we can compute the true odds ratio. So that's this true value vertical dotted line here. We then simulated having unlabeled data by tossing out the vast majority of those labels, pretending we never saw them, like pretending this was just like a bunch of unlabeled data, and instead getting predicted intrinsically disordered regions using predicted AlphaFold predicted structures. We then use the classical approach, which only looks at the labeled data, the imputation approach, which only looks at the predictions, and then our approach to compute confidence intervals for this odds ratio. And I'm going to show those on the next slide, but just remember there's two things we want from a confidence interval. We want it to contain the true value, so contain this vertical line, and we also would prefer it to be small so that it's actually informative to us. So the imputation interval in orange, and the classical is here in gray. And as expected here, the classical one is valid. It does contain the true value, but it's quite wide due to the small amount of labeled data. Whereas on the other hand, the imputation interval is really small because there were so many unlabeled sequences to get predictions for, but it doesn't contain the true value. It's pretty misleading. because of prediction error that wasn't corrected for. Okay, so prediction-powered confidence interval basically gives you, again, the best of both worlds. You get something that's valid and contains the true value because you're correcting for prediction error. And at the same time, you're getting something that's smaller than the classical interval because you're effectively increasing sample size by leveraging information from the predictions. Okay, so this is just like a quick kind of nice visual of like the benefits of this. And I'll now go into exactly how this works. So on this slide, I'm just going to give kind of a like cute conceptual diagram of like the high level feeling of how it works. And we'll dive into the technical details on the following slide. So there's kind of 3 core steps here of how prediction-powered inference works. It starts with identifying what we call the rectifier. So the rectifier is the specific notion of prediction error that is relevant for your estimand. So if you're estimating the mean value of y versus like the 80th percentile value of y versus some linear regression coefficient, prediction error is going to affect those different estimands in different ways. And the rectifier is like the specific notion of error that's appropriate for kind of correcting your particular estimand. So just to visualize this, like in this diagram here, theta star on the right is your estimand. So for example, if you're estimating the mean value of y, that's theta star, the expected value of y. And then theta f on the left is like the analogous quantity, but based purely on the predictions instead of the labels. So here that would be the expected value of f instead of the expected value of y. And theta f, of course, is usually biased due to prediction error. It's not going to be what we want to be using. And so the rectifier, which we call delta f, is that particular notion of error needed to correct this prediction-based quantity, theta f, to get to kind of the true quantity, theta star. And in this simple example, the rectifier is super, super intuitive. It's literally the bias. It's literally just the expected value of f minus y. And if you take one and subtract it from the other, then you get to theta star, which is what you want. But for other estimands that are not the mean value of y, the rectifier is going to be different. And kind of the main technical piece of our work is like basically identifying, giving a general recipe for identifying and deriving the rectifier for a very broad class of estimands that you might care about. And I'll go a bit more into that in the next slide too. Okay, so the second step is to construct a confidence set on the rectifier. So the rectifier is some population quantity that needs to be estimated. So we're going to use the labeled data to build a confidence set for that quantity, for the rectifier. And we're going to call that R. And finally, we're going to form the prediction-powered confidence set in green by basically propagating theta f through all possible values of the rectifier in the rectifier's confidence set R. So basically, the prediction-powered confidence set contains all values that result from taking theta f and correcting it with a high probability value of the rectifier. And as long as that confidence set for the rectifier, R, is valid, then the green confidence set, the prediction-powered confidence set, is also valid, meaning it contains theta star. Okay, so one intuitively nice thing that I'll point out, which I'll go into a bit more detail soon, is that the rectifier confidence set is going to be small if your predictions are very accurate. So for example, the bias here, delta F, the bias, will be 0 if your predictions are exactly equal to the labels. And so the result of that is that the better your predictions are, and the smaller the rectifier confidence set is, then the smaller your prediction-powered confidence set. Okay, so I'll turn to more kind of technical details on the next slide, but I'll pause here for kind of high-level questions about the concepts first.
All following along still, Clara.
All right, great. Oh yeah, so I did not make this illustration. I will give credit to that. to my incredible co-author, Anastasios, who I think he did this just like on an iPad, like Notability or something. But yeah, that I did not do this particular illustration. Okay, so to make what I just said concrete, I'm going to sketch out our main technical result here, which is exactly how to do this for any estimate that can be expressed as the minimizer of a convex loss, where I mean convex in terms of theta. So this, for example, could include the mean value of y, and for the mean value of y, then L theta would just be theta minus y squared. You can get quantiles of y, when L theta is what's called a pinball loss. You can get various generalized linear regression coefficients if you kind of put in the maximum likelihood expression for this argument expression. But this is just a very broad class of estimates that we can handle with this framework. There's also an extension like buried deep in the appendix for non-convex losses, which is not by any means computationally tractable, but there conceptually are lots of extensions to other classes of estimates as well. Okay, so let's think about the gradient of this loss with respect to theta. So I'm going to call that G theta. Since the L theta is convex in theta, we know that theta star is essentially under like mild regularity conditions. It's the same as the value of theta such that the expected value of the gradient G theta is 0. So our approach is going to be to build a confidence set that contains the value of theta such that this expected gradient equals 0. And the way that we're going to do this is we're first going to rewrite this expected gradient and decompose it into 2 parts. So I'm just going to split it up basically with a simple debiasing trick here. So it equals, this expression just equals the expected gradient evaluated on the predictions F minus the bias of that component. So the expected difference of the gradients when using the predictions F versus the labels Y. So now we have 2 pieces. And basically note that these 2 pieces match what we saw on the previous slide. So if I go back, so here conceptually we're dealing with kind of 2 things that we put together. One is theta F. It's a thing that's purely based on predictions. And then the rectifier delta F is like a notion of prediction error. So here again, we have those 2 pieces. The first piece is a component that's purely based on predictions. It's the expected value of that gradient when evaluated using predictions. And the second piece is exactly what the rectifier is. This is the notion of predictive error that we focus on in this work. And it's just the bias of the gradient of the loss. So the expected difference in that gradient G theta when I evaluate it with the predictions F versus evaluating with the labels Y. And so what we do is we build a confidence set R theta for this rectifier using the labeled data where we have both the predictions F and the labels Y. And you can use any number of like off-the-shelf statistical tools for doing this confidence set. You can do confidence intervals based on the central limit theorem or a number of other tools for doing kind of finite sample valid things. But the point is that we're going to be using the labeled data in this setting to basically estimate this rectifier, which is the bias of the gradient. So our main theorem looks like this. Basically, we're going to take CPP, the prediction powered confidence set, to be all values such that 0 belongs into this set. It's the prediction-based gradient minus R theta, where R theta is a valid confidence set for the rectifier. And if you just construct CPP to be exactly like this, then it's going to be valid. The true value theta star will fall within CPP with guaranteed high probability. Okay, so there's kind of 2 notes I want to make here to maybe help intuition. Remember, there's two things we're looking for in a confidence set. One is that we want it to be valid, and the second is that we would like to be kind of squeezing information from the predictions to increase sample size and effectively get smaller confidence sets. Okay, so for the first thing, this simple debiasing trick of just like separating out the component that's purely based on predictions and then this component that has to do with prediction error, this is exactly how we maintain validity. It's a very simple insight that's been used across many literatures and statistics. But that part is like this rectifier is exactly how we achieve that. And the second aspect is that the rectifier confidence set is going to be small if the predictions are good. So if my predictions F are very, very close to my prediction to the labels Y, then the size of the rectifier confidence set R theta is going to be pretty small. And then correspondingly, the size of the prediction powered confidence set CPP will also be small. So that's effectively how, like at a technical level, we're able to get smaller confidence sets with good predictions. I'll pause here for any questions.
I have a question.
So I know, so it seems like the ability to build confidence sets for the rectifier is really at the heart of this. And I think you answered very clearly why you probably want to make sure that your predictive model is not trained on the exact same data that you're using to sort of calibrate this. But it seems like there's another assumption about your labeled data needing to be distributed the same way as your unlabeled data that you're going to use the predictions for. And I could imagine a world in which, maybe we know protein structures for proteins that tends to be less disordered than the average protein. So I guess like how is that an assumption that you do have to make, and how often is that going to be true?
Yeah, that's a really great question. As in all of statistics, of course, there are some distributional assumptions here. The simplest version of the result is that yes, the unlabeled and labeled data have to be IID, or it can be like a finite population situation where like you have a database, a finite database, you randomly sample some of it to label and you leave the rest of it unlabeled. That also, we have results for that. We also have results for like if you have covariate shift or kind of mild forms of label shift. But you're totally right that you need some sort of distributional assumption connecting the unlabeled data with your labeled data. So there's actually kind of two assumptions you need to be able to properly assess and propagate that error. Like you said, one is that the data that you train the model on is not contaminating either the unlabeled or labeled data. And the second is that once you estimate the rectifier with the labeled data, the only way that can have any meaning for the unlabeled data is that if we know exactly how those two things are related. So yeah, we do need those kind of assumptions.
So are there certain types of distributional shifts that make it so you can never construct a valid?
Oh, I don't know. I guess you just need, there are various ways to, devise assumptions where like you can just propagate the error in one way or the other. So you do have to know something. I think like in practice, the one that I would be most kind of just comfortable like recommending use of this is like where you have a fixed large database and you have control over randomly selecting some of that to label and you leave the rest of it unlabeled, which is like essentially it's not quite IID, but it's like in spirit the same thing. And I think it's like a practically relevant setting. There are some other settings where you know that covariate shift is happening or you know that label shift is happening, but I think it's more typical that those are very much assumptions rather than like totally known truths. So this is definitely like an area that is becoming increasingly relevant because yeah, like you were saying, like there's lots of different databases that we want to be combining and working with together. And the stuff that has labels usually looks systematically different from the stuff that doesn't have labels. That's exactly why they're different in that way. And that's definitely a huge kind of area to look into. In the lack of knowing exactly how your distributions are related, I would probably resort to using various like sensitivity analyses to like assess kind of if exactly how much the results would differ if the distributions were different in one way or the other. But yeah, it's a great question. Are there any other questions?
I think we can keep going for the time being.
All right, great. Yeah, I should also note that, you know, this core idea of like debiasing is in the setting of having a little bit of labeled data and a lot of unlabeled data is not at all new in statistics. It shows up in a field called model-assisted survey sampling, which uses debiasing to incorporate auxiliary information into survey data. It also shows up a lot in the semi-parametrics literature. And the typical setting in that literature is that you actually train a model on the labeled data, make predictions, and then want to estimate a parameter of that same model. And so this is a little bit of a different setting from ours, but we do still continue in the spirit of these two very rich fields. And of course, the key distinction is that we're assuming a fixed model that has already been trained on separate data, as we just talked about, which is kind of an increasingly relevant setting. And this frees us from having to make any sort of assumptions on the kind of machine learning model, the details of how it's trained. It really can be anything. It doesn't even have to be a machine learning model. Any sort of thing that spits out predictions, like simulations, MD simulations, kind of biophysics-based predictions. The prediction engine could be like, call Jen and ask her what she thinks, assuming her thought process for making the prediction doesn't change. But it really can be anything. It doesn't, like, we make no assumptions at all on how these predictions F are produced. And that's definitely a deviation from the semi-parametrics literature and a lot of these other kind of older statistical areas that focus a lot on the specifics of the model to do the estimation. Okay.
We have some questions. Hang on.
Yes.
I have two questions. And feel free to punt if it's useful. But first question is, what is this probability with respect to? So this looks very similar to like kind of conformal prediction type. Problems in conformal, you have like some like probabilities over like X, Y, and like recalibration data. What is this probability with respect to?
Yeah, so that's a great question. So this probability is like the same kind of probability that like, well, okay, so it's the probability with respect to the labeled data and unlabeled data. So like in the setting, for example, if they are IID, then it's just with respect to drawing the labeled data and unlabeled data IID. At a higher level, like this is the same kind of probability of how people compute confidence intervals in a kind of classical statistics approach in basic science. So it's like a frequentist notion of probability with the data. And it's interesting to note the connection to conformal prediction. There's some similarities, but the goal of these two methods is really quite different. The goal here is we focus on this setting that shows up a lot in basic science. Like there's some property of the data distribution, some functional of that data distribution that a scientist wants to put confidence intervals or a p-value on, like testing, like they want to know the mean height of, I don't know, some bird species or whatever in some population. Whereas the goal in conformal is to put a confidence interval around one specific like data point. So you can come up with like convoluted ways of like using conformal to kind of get at this problem, which we did for the sake of publication review, and that doesn't work out well. But yeah, there are some kind of aspects that are similar.
And as a follow-up, so is it similar though? I mean, can you get some sort of bound like conditional on the label data that you do see? What is this probability of theta star being inside of this CP?
I see.
So
Yeah.
I think in the conformal prediction literature, there is like a result, which is like this probability distribution is like a lower bounded stochastically by like a beta distribution or something like that. Does that result kind of translate into this? Is there a similar result in this case?
That's an interesting question. I haven't thought about that, to be honest. I don't think so, at least not with the tools that we used. But I can totally see how that would be very relevant if one were to use this in practice and like you have this like precious baby gold standard data set you want to be able to use for sure. So yeah, that's a great question. I haven't thought about it. I don't think there is such a result, but it's worth thinking a bit more about.
I think that'd be really interesting to think that, just because like, what if this probability, like the probability alpha, where this doesn't hold, is just all concentrated on some like really bad.
Sorry, I can't quite hear.
Oh, sorry.
Oh, that's great. Okay.
Yeah,
I heard you say the probability the alpha error was all concentrated on like some certain bad. I'm assuming you meant certain
some bad labeled data.
Yeah. In the case where they're IID, I don't. I can see how like in the IID setting, this is probably one of those cases where you can construct like contorted like pathological cases where that would be the case, but it's like it's really not gonna happen in practice. But yes, TBD, it should be kind of explored further for sure.
I think we can keep going.
Okay, cool. Oh yeah, so yeah, just these are just a few more examples that have kind of shown up either in our paper or in discussions with like for potential collaborations. But this can capture a huge range of different quantities you might want to be estimating, whether it's frequencies of certain events or associations reflected by regression coefficients in ecology, biology, astrophysics. This all sorts of fields can kind of benefit from this.
Are you using it at Genentech?
Oh, am I? Yes. I'll say a bit briefly about that later. Very briefly about that later. I just want to kind of highlight here that the code for these methods is not complicated. It depends a bit on exactly what kind of confidence intervals you want to construct, like whether you want them to be asymptotically valid versus finite sample valid, meaning valid for any amount of data. But this would be the example for the code for prediction powered confidence interval for the mean value of y. It's like 10 lines of Python. This is for a quantile of y. It's a bit more involved because the rectifier in this setting is a bit more involved. But it's still within 30 lines of code. So these are very much practical kind of algorithms that don't require a lot of overhead to be implementing instead of the classical or kind of imputation approaches. Okay, I'll finish up by just quickly showing the framework in action on a few different data sets. So here I'm just revisiting that Blue-Dow et al. Paper that I was very much inspired by to kind of think about this problem. So here, just showing the same kinds of experiments I previewed earlier, where we're constructing confidence intervals for an odds ratio between intrinsically disordered regions and PTMs. And there's lots of different kinds of PTMs. So we did this for a few different types. On the left here is a type called phosphorylation. On the right is a type called ubiquitination. And so in both cases, like qualitatively, we kind of have a similar result where so the true value of the odds ratio that we want to be capturing is the vertical orange dotted line. The gray is the classical interval that only uses a small amount of labeled data. The orange is the imputation interval that uses an enormous amount of predictions but doesn't correct for a prediction error. And the green bars are kind of the prediction powered confidence intervals. And we just showed
Just
to
interrupt, I've been looking at the central screen where I have not been tracking. So for those of you who've been doing the same, you can see it on the side LCD screens. I was wondering what you were talking about for a while, which is not your fault, and you can't see the screens. I just want to make sure that whoever else was doing what I was doing looks at the other screens now. Okay.
Okay, yeah, hopefully what I'm talking about is clear. But yeah, so the orange arrow that you see on both plots is like, I'm just trying to indicate the imputation interval, which is super, super small because of the enormous amount of unlabeled data that we have predictions for. It's very small and very confident, but wrong because of prediction error. And I'm showing a few different trials of the classical intervals in gray and the prediction powered intervals in green to kind of reflect that randomness that we just kind of talked about. And then, yeah, so we see this trend. I'll just mention again, this trend of the prediction powered ones being smaller than the classical ones. And on the bottom plots here, I'm just showing the width of those, the average width of those intervals for different amounts of labeled data. So the x-axis on the bottom subplots, the x-axis is little n, which is the amount of labeled data. So for a large range of amount of labeled data, we're always seeing that the prediction-powered intervals are smaller than the classical ones while being valid. Though that kind of that benefit is particularly extreme for very small amounts of labeled data. Which kind of intuitively makes sense because when you have very little unlabeled data, our method is able to leverage all the information from the unlabeled data. And that benefit gets smaller and smaller as you get more and more labeled data. But we're always kind of writing, you're basically like this green line is also always writing beneath the gray line. I'll just show kind of two more brief vignettes on this slide. So on the left is this task of doing gene expression analysis with predictive transformers. So here we have a population of regulatory DNA sequences called promoters, which are naturally found in yeast. And for a relatively small number of those sequences, we have experimental measurements of this real value gene expression level caused by the promoter. And for like tens of thousands more promoters, we have predicted gene expression levels from a kind of state-of-the-art predictive transformer model. And the goal here is to basically estimate different quantiles of the gene expression level in this population. So in the left subplot, there is the 0.5 quantile or the median gene expression. On the right is the 0.75 quantile. Again, the vertical orange dashed lines are like the true quantity that we want our intervals to be capturing. And in both cases, the imputation interval is very small, doesn't cover the true value. The classical one does cover the true value, but is often bigger than the prediction powered intervals that we have. And again, we see this this trend of like we are always getting smaller and more informative confidence intervals regardless of the amount of labeled data. And finally, this is an example I really like. It's prediction-powered galaxy counting. So the setting here is that we have a bunch of galaxy images from the Sloan Digital Sky Survey. And for a very small number of them, we have kind of expert astronomer labels with the label of whether or not the galaxy is a spiral galaxy. And we also have predictions of whether or not it's a spiral galaxy from a computer vision model. And the goal here is to estimate the frequency of spiral galaxies in the local universe. I'm not from this field, so I can't really comment on the implications of this. But it was a task that we found in a few papers. And again, we see this pattern of the imputation, confidence interval being very, very small, but invalid. The classical intervals in gray being valid, but larger than what we would get with the prediction powered interval. Right, and we see that same trend of always being smaller than the classical interval. And yeah, that's kind of a wrap. Just as a quick summary, this is a method for using both label data and predictions to estimate scientific quantities of interest. I didn't explicitly comment on this here, but because of a duality between confidence intervals and p-values, you can use the same techniques to get prediction-powered p-values and do hypothesis testing with predictions as well and get like valid p-values. You can use any machine learning model. Again, we didn't make any assumption about what was providing the predictions F. It doesn't even have to be a machine learning model. It's just like anything that spits out some approximate quantity that you want to be leveraging information from. The results that we get are statistically valid, and you can kind of pick and choose whatever kind of validity you like the most. So if you want asymptotic validity, you would just use asymptotically valid confidence intervals for the rectifier. If you want finite sample validity, you would use finite sample valid confidence intervals for the rectifier. So it's very flexible in that notion. And we also have this very nice, like intuitively pleasing thing where the better your predictions are, the smaller your resulting confidence sets. So this leads to a very nice paradigm of like, the better you can make your predictions, the more information you can effectively feed into your current study, the more you can basically increase sample size. Yeah, and so I'll just mention briefly that I'm currently really interested in, on the methods side, leveraging related kind of ideas for doing evaluation in the setting of machine learning guided design, which is one setting where we might want to design new proteins, new biological sequences that have various new functions. And because of the cost of validating such things in the wet lab, it'd be really nice to be able to estimate how they're going to be performing a priori. And this is certainly a setting where we might be able to make predictions for the labels of those designs, but there's like, without actually going through the expensive experiment, you don't actually have labels for those designs. So we can use similar techniques from prediction-powered inference to kind of tackle that task. There's also extensions to various tasks in causal inference I'm really excited by. And on the application side, this is really totally open like field to kind of explore. I think there's a lot of really big questions you can ask now with this tool, now that you have hundreds of millions of AlphaFold predicted structures, or there's lots of databases of like predicted patient phenotypes, medical imaging databases we can leverage predictions from to make conclusions. So there's lots of questions now that I think that can only be answered with this new paradigm of doing science. So I'm really interested in thinking more about applications and those kinds of collaborations as well. Okay, with that, thank you guys again for your time and allowing me to share this. Thanks again to Jen and DT for organizing and putting this all together. These are my colleagues and co-authors that provided guidance on this work. Yeah, and I'm happy to take any kind of further wrap-up questions now, or feel free to contact me offline too.
You comment more on, you've said a few times, if you want asymptotic versus finite sample guarantees, like tell me what I should want, Clara, and tell me how much harder it is to get.
Oh, I feel like I'm wading into a potential like philosophical fight here.
And I asked the right question.
Sure. Well, I mean, if you were to pull a random paper from basic science, broadly speaking, being written today. Those confidence intervals and p-values in a randomly selected basic science paper today are going to be based on central limit theorem. So they're going to be asymptotically valid. So it would be totally within the realm of just how science is being done today to do asymptotically valid confidence intervals and p-values. And in fact, that is what I would probably recommend in practice, because they're actually much easier to compute because you get to leverage closed form expressions of Gaussians. And practically,
could you comment slightly more on like the time complexity or like are the computations scaling much worse? Like the code is uglier. Like what happens?
What happens if you do go for finite
guarantee versus the asymptotic?
Yeah. The field of kind of like deriving finite sample valid confidence intervals is like actually still currently a very active field. So it's a much kind of newer statistical paradigm in some ways, at least in terms of its popularity. And so there are increasingly kind of sophisticated techniques for doing it. Maybe to put it at a very kind of like practical low level, many of them involve, in theory, iterating through the real line and checking each value and doing some sophisticated computation with it. So you're going to end up having to approximate that by getting a grid of candidate values, checking each one. Whereas for asymptotically valid confidence intervals, you basically estimate the parameters of some Gaussian, and then you add and subtract a sigma of that Gaussian, or the standard deviation of that Gaussian. So like many orders of magnitude faster, typically. And at least in the applications that I've kind of played around with, for typical amounts of like labeled data and unlabeled data that I've found in various applications, like the asymptotically valid intervals just tend to be valid in practice, at least for the numbers that I've seen. So I would probably advise doing that in practice.
Thank you.
Hey, Clara. Thanks for the talk. I'm curious if you have any thoughts on how these tools could potentially be used for experimental design. Like, let's say you have a new problem that you want to estimate some quantity for, and you have some experimental budget and some computational budget. Is there any way to modify these types of tools in order to understand how I can get the best confidence interval for my quantity that I care about?
Okay, sorry. First, is that David?
Yeah.
Oh my gosh. Okay, hi David. I couldn't quite tell because you're so small on my screen. But it's really good to see you. Okay. Secondly, so do you mean like you have some budget for labeling?
Yes, experimentally and computationally.
Okay, so Tiana Zernich, who is one of the authors on this work, she's now a postdoc at Stanford with Emmanuel Candace. She just, as of a few months ago, I think, released a paper doing basically exactly what you're saying. I think it's titled like active statistical inference, where the idea is like, instead of doing active learning where you have a budget for picking which things you want to label for the sake of training a model, it's like you have a budget for picking labels for the sake of doing inference. And it's a very direct follow-up to this work, and I think it exactly addresses your question. But the short answer is yes, she figured out a way of doing exactly what you're saying.
Thanks for the talk, Clara. I was wondering if you could comment on if there's any relationship between something like this and bootstrapping, and because those things also in some ways seem like they could be compatible.
Oh, okay. Yeah, I see. Like, you mean, like, is there some way to either use the bootstrap to, in this kind of sub problem of doing confidence sets for the rectifier. Yeah, so
exactly.
so the core contribution of this work is not about how, in some sense, it's not really about how to construct a confidence set. It's more about like how to combine predictions with some notion of prediction error to get rid of that prediction error when doing estimation. And then we use like, you know, construction of confidence sets as a sub problem that can be, you can use any kind of off the shelf tool for doing that. And so if you wanted to bootstrap your way into getting those confidence sets, you could do that. I'm not a huge expert on like the exact nature of the validity of bootstrapped kind of intervals, and I think the conditions for that are a little bit more delicate than what we've dealt with here. But so long as one is okay accepting the potential kind of vagaries of that, then yeah, I think that would be one way to go about doing this too.
In order to respect the time of the staff of the Simon Institute, especially on a Friday, we're going to have to wrap it up. But first, let's thank Clara for a wonderful remote talk. And then I want to thank all the speakers and especially the people in a room on a Friday at almost 5 o'clock. And I want to thank Omid, who's been helping us a lot back there in the box, and all the Simon staff who can't hear me because it's such a fantastic place to host a meeting. And so DT and I are very grateful to have done it. So I guess that is the end of hopefully version 1, but we shall see, of our AI science workshop. So thanks again, everyone. And bye, Clara. We miss you.
Yeah, see you. Thanks, guys.